Alrighty, this is my presentation. Say sayonara to sialidosis type 1. Now for this project, we were to find a rare disease from a very long list of rare diseases and study about it, learn a little bit more about it, and then think of ways that we could help treat this disease. So for my rare disease, I chose sialidosis. Now there are two types of sialidosis, and I chose sialidosis type 1. Now, sialidosis type 1 is a lysosomal storage disease where a mutation causes a deficiency of NEU1, and I will get to that, what that is later. Type 1 is the less severe version of sialidosis. It can be characterized by the main symptom of cherry red macular spots, and other symptoms include myoclonus, and there can also be more neurological symptoms. There also can be, some ear issues, some hearing issues, and a lot of muscle issues. So this disease, the most, leads to bad vision impairment, and it is inherited in an autosomal recessive pattern. Now, based on where you are in the world and different regions of different countries, it has been found that there are a lot of different symptoms for a lot of different people with this disease. Now, for our images here, there are some examples of our cherry red spots in the macula. So at my pointer here, you can see them, and all different types of photos here, even down on grayscale. It's basically just like a big spot blocking the retinal function by overpigmentation caused by this lysosomal problems, which then can cause that vision impairment. So the functional protein that we are going to focus on for our project here is sialidase 1. So sialidase 1 is the functional protein encoded by NEU1. It is also called lysosomal neuraminidase. They also call it NEU1 sometimes, so throughout the presentation you may see it put as both, but NEU1 is also the gene that encodes it. So sialidase functions as a catalyzer in removing parts of sialic acid from various glycoproteins and glycolipids. It is also part of degrading sialoglycoconjugates, which are sugar chains modified by sialic acids, which all takes place in the lysosome. So really what it does is it comes in, it marks and cleaves off this sialic acid from these different glycoproteins and helps degrade that part of them so they can be degraded farther. And there's not an excess of them, especially in the cell. So when it's mutated, it is unable to cleave those sialic acid chains from the glycoproteins and glycolipids, which causes an accumulation of that sialic acid in cells, which can be really bad for them. So the structures, I have this picture for us to focus on here. These are our BNR domains. So they are repeats 1 through 4 are found in sialidase 1, and they are also found in most neuramidases, and they are used to cleave off that sialic residue. So this is really the main domain that is going to function and cleave off that sialic acid, which is really the whole purpose of that protein. And so they are a huge part in the degradation of toxins within the lysosome and keeping those toxins out of the rest of the cells. So of course, for here we just have those pictured. This is the secondary structure of those domains. Again, they're called the BNR domain. So now we're going to go ahead and we're going to focus on the signal pathway, how this protein is getting from where it's made all the way to the lysosome where it should be. So it follows a mannose-6-phosphate pathway. So when it's not mutated, over here in this little illustration, I would almost call it, I have made. So we have the nucleus here. So it's going to go ahead and send out that information about how that protein needs to be formed inside of that endoplasmic reticulum. Once it's formed in there, it's going to go ahead and travel along the microtubule towards the centrosome there and towards the Golgi. It's going to be carried by a dynein in a COP2 coated transport vesicle. That's protein there in the middle. It's going to be transferred to the cis Golgi. Once it gets to the cis Golgi, it's going to be marked with an mannose-6-phosphate marker. Now this marker is going to tell us that it needs to be brought to the lysosome. So once it travels through the Golgi and it is in this last part of the Golgi, the trans-Golgi network, it's going to be bound then to an M6 receptor, so mannose-6-phosphate receptor, and then it's going to go ahead and be coated by clathrin in this transport vesicle, and that's going to be moved by kinesin towards that lysosome, which then it will bind with the lysosome and it will go on in there. The vesicle will go away, the clathrin will go away, and the protein will go into the lysosome and do as it's supposed to. And it'll function in the lumen, its normal function. So when they talk about the mutated version, they just kind of talk about how it's normally supposed to work. There has not been a whole lot of research on exactly why the NEU1 is having a deficiency, and they can't really figure out exactly where it is being lost along this pathway. So it's just assumed that along the pathway, the sialidase 1 NEU1 is being lost somewhere along this pathway. Now to look at the cells and organelles that are affected in our disease. So for organelles, the cellular location of our disease is the lysosome. So in a healthy lysosome, lysosomes have a usual function of breaking down excess materials in the cell to keep the cell healthy. So with the protein sialidase 1, the lysosome can break down those glycolipids and glycoproteins, like we said before, that are left in the cell that aren't used, and they identify those by that sialic acid, and it goes ahead and it breaks that off, and it breaks those down, and then it's going to be removed out of the cell. Now, in this mutated version in the lysosome, the mutation in the sialidase 1 will cause an inability to break down those certain glycocomponents or molecules, as you could call them, and this will end up with an accumulation of sugars with that sialic acids still on, as the sialidase cannot degrade them. So toxins are going to accumulate in the cell, which are going to cause those symptoms that we had listed before, with the bad vision impairment, bad hearing impairment, some muscle issues, some neurological issues. Those are all going to be caused when this lysosome can't function properly. So decreased levels of this sialidase 1 and NEU1 is also going to cause increased lysosomal exocytosis. So it's going to be increased shedding things out of that cell, out of the membrane, into the rest of the body when they're not fully broken down, which also can cause issues. So our cells affected are the fibroblasts. So these fibroblasts turn into pigment epithelium in the retina of the eye, which is those cherry red spots we saw from before. So this occurs by using induced pluripotent stem cells. In this research that I found, this is how they did it. And these cells have a normal lysosomal function usually, so they have perfectly normal lysosomes. They function exactly how you want them in the body. But when we have the mutation in sialidase 1, they do not work correctly, and it cannot rid that extra sialic acid, causing the accumulation in those pigment epithelium. So when you have that accumulation, you get that overexpression of the color, so it causes overexpression of that color in the eye, which causes almost like that blind spot, like I said, which causes the bad vision impairment. So we've learned all about this and how it can affect people and how it works in the body, but the most important thing we need to know about rare diseases is that they don't really have treatments. And so with no cure and limited treatment options for as many rare diseases as you can think of, we really need to focus on research and ways we can treat them, and sialidosis is no different. They don't really have any treatment options, and they don't really know how to go about treating it. So with the research I did, here are a few that I found. So for option 1, betaine, this is a special kind of amino acid. It's not one of the normal ones. Amino acid isn't exactly a great name for it. It does take glycine, the amino acid, and it adds 3 methyl groups onto it. And so this can be found in foods we eat like spinach and beets. So it is a naturally occurring thing that you can find in your environment. It has been found that if you increase it in the body, you're going to find increased levels of NEU1 activity even when sialidosis is present. So it could be taken as a dietary supplement for people with sialidosis to hopefully increase the lysosomal activity of that cell to keep a normal level of lysosomal activity. So let's talk a little bit more about betaine. So the exact mechanism is not understood. They don't fully understand exactly how this works with sialidosis type 1, but they have found through research that when you give it in excess, it raises the activity levels of NEU1 even when it's been mutated. So an increase in that activity would allow for proper lysosomal function, which is going to prevent those symptoms from being as severe and can hopefully give those people a little bit of relief. Now, our second treatment option that I found was protective protein capsin A, or called PPCA for my presentation. And it is thought to increase the activity of NEU1 also. So when used in excess, it was found to help patients with sialidosis type 1. They have done some research here. So it could be taken in excess with that betaine, and it would help to raise those levels. So for protective protein capsin A, due to sialidosis being a deficiency of that NEU1, causing the excess could, help us bring a little bit more in and its activity levels up, which could help. So anyone needs protective protein capsin A for its proper function in the lysosome. It helps with protein folding, it's catalytic activity. It's really just needs to be together for the NEU1 at least to get going, to get started, for its activity to begin. So due to this effect with it, if we increase this, it has been found that it can kind of counteract the mutation and kind of just get normal levels of NEU1 working in the body, which could help lower those symptoms of sialidosis type 1. So for a summary of these, how do we achieve a little relief? So if we were to take in combination both increased levels of PPCA and betaine, we could find that the NEU1 activity would raise and that there would be not as much symptoms showing, or at least there wouldn't be as large of issues, which could maybe give some people some relief. Now, it's definitely not a permanent solution, as you can't just keep pumping these things in and hopefully that it will just always be fine. We want to find a more permanent solution, maybe a way to get around this to cure it, so obviously more research needs to be done. But this is just a start as something that could possibly help these patients get a bit of relief. And here is my works cited for you. Thank you for listening.